So InTraSeq is a technology whereby we can in fact look at the single level, the signaling components within the cell and combine it together with single cell RNA-seq.
InTraSeq is going to find new biology in your sample and things that you could never measure at the single cell RNA level, especially post-translational modifications. And it just opens kind of a whole new black box within your single cell data set that is going to be so much fun to interpret and analyze.
So far, signaling has always been done at the population level, where you grind up the cells, do either Western blot and IP and see what the phosphoproteins are there. But it is the sum of all the cells, and it may not be total truth, because if each cell is different, this is what we learned from single cell RNA-seq, and each one of them cluster and have a function.
These intracellular proteins get phosphorylated and they drive the machinery within the cell. And this in turn tells the cell whether to start transcribing or not. InTraSeq offers a way to measure these things and quantify them along with the RNA, so you can look at the whole signaling pathway within individual cells. And if you have a complex sample, that also can give you a new perspective, because more often than not, different cells respond differently to different treatments. So looking at signaling pathways and looking at what transcription machinery is activated in different cells could be very beneficial to your work.
So there was a lot of work that was done actually in my lab, and putting together, not only running some of the InTraSeq antibodies and components, but also computationally how to combine, because signaling happens within minutes to an hour, but the transcription happens in days, hours and days. And then to combine what happened in 10 minutes and what impact it has on the days and function later is actually an interesting problem which needed to be solved. And I think InTraSeq is going to be addressing that problem per se.
There's so much collaboration that went into this, starting from a concept idea, having different research groups work on this together so people can use InTraSeq in their single cell assays and learn new biology in their experiments. Yeah.
It's going to be actually a revolutionary technology that will probably allow us to look at many of the important components of cellular signaling at the single cell level.